Good day, future doctors. So this will be our lecture on histology of the lymphatic system. Before we'll discuss the different tissues and organs that belong to the lymphatic system, let's have a review on the different fibers present in our tissues. In tissues or organs that need to be tough and strong, they should have abundant collagen fibers. For organs that can stretch beyond their original size, they must have abundant elastic fibers. But for organs that contain a lot of cells, just like the organs of the lymphatic system, the cells there are supported by reticular fibers. So from this point on, I want you to remember that all of the tissues and the organs that we will be discussing under this chapter are abundant in reticular fibers. This is the microscopic picture of a lymph node. Appreciate that it contains a lot of cells. So therefore, this organ should be rich in reticular fibers. Before we'll discuss the different organs that belong to the lymphatic system, it's very important to be reviewed on the terms parenchyma and stroma. When you say parenchyma, these are the active cells of the organ. So in terms of the lymph node, the spleen, and other lymphoid organs, the active cells are the lymphocytes. And you know very well that T cells and B cells are lymphocytes. So therefore, they are the parenchyma of the lymphatic system. When you say stroma, these are structures that support the parenchyma. So what structures in the lymphoid organs provide support and protection to the T cells and B cells? Take a look at the picture of the lymph node and then refer to the 6 o'clock position of the photo. Please look for that structure that is labeled as capsule. The capsule is the protective covering of the lymph node. It is there to provide support and protection to the T cells and B cells. So therefore, capsule is one of the structures that belong to the stroma of the lymph node. And then please do appreciate that at some points, the capsule will invaginate into the lymph node, forming now the structures labeled as trabeculae, plural, trabecula, singular. So the second structure that belongs to the stroma of the lymphoid organs is the trabecula. You have learned from histology that if an organ contains a lot of cells, the best fiber that would support these cells is reticular fiber. So that makes reticular fiber as the third structure that belongs to the stroma of the lymphoid organs. Stroma supporting structures of the parenchyma of the organ, and the stroma includes the capsule, the trabeculae, and the reticular fibers. We will prepare a thin section of this lymph node and we will view it under the microscope, and this is how it will look like. At this point, we will try to identify the structures that belong to the stroma of the lymph node. We have here the protective covering, which is the capsule, the extension of the capsule, which is the trabecula or trabeculae, and you know very well that if the organ has a lot of cells, the best fiber that would support them is reticular fiber. The lymphatic system is composed of lymphatic vessels and lymphatic tissues or organs. We will first discuss the lymphatic vessels. You have here in this picture the red-colored arteriole and the blue-colored venule. Between these two blood vessels, you have there the thin-walled capillaries. The capillaries are so thin that every time the blood will flow through their lumen, the blood will exert pressure on their walls, causing some of the fluids to leak out, and that will somehow help in the diffusion of nutrients. But the thing is, fluids will accumulate in the tissue through time because the blood will continuously flow through the capillaries. And we cannot afford for that to happen, or else the organ will swell because of the accumulation of the leaked fluids from the capillaries. That is now solved by the presence of those green-colored vessels, and we will name them or refer to them as the lymphatic vessels. The fluids that have escaped out from the capillary will now be called as the lymph, and what will collect the lymph are these green-colored lymphatic vessels. The functions of the lymphatic vessels is to collect the lymph that has leaked out from the capillaries. But aside from that, these lymphatic vessels can sample the antigens present on the tissue or organ. What do we mean by lymphatic vessels sampling the antigens present in the tissue or organ? Let's assume that the organ supplied by these blood vessels is currently infected. What will happen is, once the lymph is formed, the lymph will cause all of the microorganisms to flow through it, and as soon as the lymph will be drained into the lymphatic vessels, the lymph will carry along with it the microorganisms. So the microorganisms now will flow along with the lymph inside the lymphatic vessels. And do you know what will happen to the microorganisms in the lymph? They will eventually be trapped and killed in the lymph nodes. So this picture is showing us that the microorganisms that came from the organ where the lymphatic vessels originated are now trapped, filtered, and being killed by the cells of the lymph node. And since the cells of the lymph node will proliferate in the presence of these microorganisms, the proliferation will eventually cause the lymph node to become swollen. That's why if you will encounter patients in the future with lymphadenopathy, one of the things that you should bear in mind is the possibility of infection. So, let's say you have a patient with cervical lymphadenopathy. What you need to do is to evaluate the origin of the lymphatic vessels draining into the swollen lymph nodes. So in the case of cervical lymphadenopathy, you have to check for possibility of oral, dental infections, and possibility of tonsillitis and pharyngitis. This is just to show you that the lymph collected by the lymphatic vessels or lymphatic capillaries will all pass through the lymph nodes wherein the microorganisms are trapped and killed. So the lymphatic system now is composed of vessels, tissues, and organs that work together to circulate a colorless watery fluid, and we call that as the lymph. And later on, I will make you appreciate that these organs, tissues, and vessels will also function to put back the lymph into the circulatory system. Anyways, the lymph was derived from the fluids that have escaped out from the capillaries, and capillaries are part of the circulatory system. Aside from circulating the colorless watery lymph, these vessels, tissues, and organs also function to monitor body surfaces and internal body compartments to detect presence of antigens. If antigens are indeed present, then these organs and tissues will create immune responses against them. The parenchyma of the lymphatic system includes the T cells and the B cells. Majority of them are circulating along with the lymph, 70%, while the remaining 30% are found within the secondary lymphoid tissues. Please recall that T cells and B cells are produced and they mature in the primary lymphoid organs. Once they will reach maturity, they will now leave the primary lymphoid organs and stay in the secondary lymphoid organs. Again, the lymphatic system is composed of vessels and tissues or organs. At this point, we will try to identify the different tissues and organs that belong to the lymphatic system. We have number 1, the spleen. We have the thymus. And we have the lymph nodes. Also included is the bone marrow because bone marrow functions to produce the T cells and B cells. But there's one thing I want you all to take note. What is unique about the organs and tissues that belong to the lymphatic system is that some of them are found in organs that belong to another body system. And we collectively call these tissues as Mucosa-Associated lymphatic tissues. So I want you to take a look at this group of lymphatic tissues here that are found in the appendix. Appendix is part of the digestive system, but there are lymphatic tissues found in the appendix. Therefore, these lymphatic tissues will now be referred to as mucosa-associated lymphatic tissues. There are two types of mucosa-associated lymphatic tissue, MALT. The first type of which is the bronchus-associated lymphatic tissue or BALT. These lymphatic tissues are found in the organs that belong to the respiratory system. So take a look at the cut section of the bronchiole. Can you appreciate that below the epithelium of the bronchiole, you have there group of cells that is labeled as lymphoid tissue. So this group of lymphocytes here will be referred to as bronchus-associated lymphatic tissue. And they are examples of mucosa-associated lymphatic tissue. The second type of MALT is gut-associated lymphatic tissue. And this type of tissues are found in the organs that belong to the digestive tract. So the lymphatic tissues that are present in the appendix belong to the gut-associated lymphatic tissue. These are the examples of the mucosa-associated lymphatic tissues. We have MALT in our tonsils, and we have three types of tonsils, the lingual, palatine, and pharyngeal tonsils. We also have MALT in our ileum, and we call that as the Peyer's patches. And we also have MALT in the appendix. This is the palatine tonsil. As you can see, this is supposed to be part of the digestive system, but you have there lymphatic tissues. So therefore, this is an example of MALT, mucosa-associated lymphatic tissue. This is the lingual tonsil, and the lingual word means tongue, and tongue is part of the digestive system. These are the Peyer's patches of the ileum. So since the ileum is part of the gastrointestinal tract, we will refer to the Peyer's patches as part of the gut-associated lymphatic tissue. And these are the lymphatic tissues that we can find in the appendix. You have learned from the introduction to immunology that lymphoid organs can be classified as primary or secondary. When you say primary lymphoid organs, these are organs that serve as site of production and maturation of the parenchyma of the lymphatic system. We are talking about the T cells and B cells. T cells are produced in the bone marrow and they mature in the thymus, while the bone marrow will serve as the site of production and maturation for B cells. And once the T cells and B cells will become mature, they will immediately leave the thymus and bone marrow, and they will now go to the next set of lymphoid organs. And these are the secondary lymphoid organs. It is in these lymphoid organs where the mature T cells and B cells are stored. It is also in these lymphoid organs that they will wait for their encounter with their antigen. And once they will encounter their antigen, they will proliferate and differentiate to create immune responses against the specific antigen. So the other organs that I have mentioned will belong to the secondary lymphoid organs. And what are these organs? The tonsils, the lymph nodes, the spleen, and the lymphatic tissues that belong to the MALT, mucosa-associated lymphatic tissue. At this point, we will be discussing how do T and B cells circulate in the lymph. Let us start with this picture. We have here the red-colored arteriole, you have there the blue-colored venule, and then between them is the capillary. You also have there the green-colored lymphatic capillary. Please take note, lymphatic vessels or lymphatic capillaries are described to be blind-ending blood vessels. What do you mean by blind-ending? The artery is continuous with the capillary is continuous with the venule, and that's a closed system. Take a look at the lymphatic capillary. it is not connected with another vessel. That's why it's described to be blind ending. So lymphatic vessels are networks of blind ending capillaries which function to collect the lymph, the fluid that has escaped out from the capillaries. Aside from doing that, they are also known to sample the microorganisms and antigens present in the tissue or organ. The thing is, microorganisms have large sizes. Antigens, most of the time proteins, have high molecular weights. So how will these lymphatic capillaries bring these microorganisms and antigens into their lumen so that they would circulate along with the lymph? The answer is, these lymphatic vessels are provided with flap-like mini valves. Please take a look at the red arrow. It's showing us that lymphatic vessels terminate blindly. They are not connected with other vessels. And aside from that, the red arrow is showing us that in the walls of the lymphatic vessels, you have there the presence of flap-like mini valves, which can just open and allow the lymph that is carrying the microorganisms and antigens to flow from the tissue towards the lumen of these lymphatic vessels. You know very well that the lymphatic vessel will carry the lymph and as well as the antigens and microorganisms towards the lymph nodes. Lymph nodes have two important portions. You have the concave side, you call that as the hilum, and you have there the convex side. And based on what you have learned, the lymph will enter through the convex side of the lymph node. So therefore, the lymphatic vessels that are found on the convex side of the lymph node will be named afferent lymphatic vessels, because whenever you use the term afferent, it means it is introducing something into an organ. So the lymphatic vessels found on the convex side of the lymph node are referred to as afferent lymphatic vessels, while lymph exit through the hilum. So therefore, the lymphatic vessels that you can see in there should be referred to as efferent lymphatic vessels. There are two ways on how lymphocytes can enter the lymph nodes. The first one is via the afferent lymphatic vessels found on the convex side of the lymph nodes. Some of the T cells and B cells that had entered the lymph node will remain in the lymph nodes. Remember, 70% of them are in the circulating pool, while 30% are found in the secondary lymphoid organs. While some of them will exit the lymph node via the efferent lymphatic vessel present on the hilum of the lymph nodes. The thing is, the lymph that will enter the afferent lymphatic vessel at the convex side of the lymph node will be carrying microorganisms. And these microorganisms now will be filtered as they flow through the lymph node. So the lymph node will make it a point that once the lymph will leave the lymph node via the efferent lymphatic vessel, that lymph node is already antigen and microbe free. Do you know what will happen? These efferent lymphatic vessels will eventually drain into the right lymphatic trunk. And take a look at the picture. This right lymphatic trunk will eventually drain to the right subclavian vein. So the thing is, the lymph now is returned back to the circulatory system. So this is just to show you guys that the efferent lymphatic vessels will drain into the lymphatic trunk, and the lymphatic trunk will drain into the subclavian vein, returning now the lymph to the circulating blood. That's why I have to show you this slide again, because I mentioned a while ago that The lymphatic system is composed of vessels, tissues, and organs that function together to move that colorless watery fluid that we call the lymph. And aside from that, they also function to return back the lymph to the circulatory system, which we have already emphasized a while ago. The second way on how lymphocytes can enter the lymph nodes is via the high endothelial venule. But before that, I want everybody to take note that the lymph node is an organ, and organs in the body have their own blood supply. So that's why expect that this lymph node would have its own arterial blood supply. And where is now that high endothelial venule? It is found on the area within the lymph node where the artery will drain towards the vein, or the arteriole will drain towards the venule. This is the arteriole supplying the lymph node. This is the venule. The area where they will intersect is called the high endothelial venule. And this will serve as the point of entry of lymphocytes into the lymph nodes. What makes this high endothelial venule different from your typical blood vessel is that instead of being lined by simple squamous epithelium, the high endothelial venule has a simple cuboidal epithelium. This is a thin section of the lymph node, and then take a look at the high endothelial venule. It's very obvious in the picture that it is lined by simple cuboidal epithelium. The entry of the lymphocytes into the lymph node via the high endothelial venule is called the lymphocyte homing. And this homing process involves three important steps. Homing is activated especially if the lymph node has filtered microorganisms or antigens. So the T cells and B cells specific to those microorganisms and antigens will be recruited from the blood circulation towards the lymph node, and that is made possible by the high endothelial venule. So you have here in this picture a cross-section, thin section of the lymph node. Shown here are two high endothelial venules. And then I want you to take a look at the arrow that is labeled homing. It's telling you that the lymphocyte should transfer from the blood circulation towards the lymph node. There are two patterns of arrangements for lymphatic tissues in the organs where they can be found. They can be in diffuse pattern, or they can be in a compact, well-circumscribed pattern that we call as the nodule. The lymphatic tissues are in diffuse pattern if the lymphocytes will accumulate with no clear boundary. And most of the time, these diffuse lymphatic tissues are found in some of the organs that belong to the gastrointestinal tract, genitourinary tract, and respiratory tract. And you can always find them below the epithelium. These finger-like projections are the villi of the small intestine. Please take a look at the simple columnar epithelium of the small intestine. And below the simple columnar epithelium of the small intestine, you have their accumulation of lymphocytes. And these lymphocytes are arranged in diffuse pattern. Take a look at the lymphocytes found below the epithelium of this bronchiole. They are arranged in diffuse pattern. But if the lymphocytes are organized in compact, somehow circumscribed structure, then we will call or refer to that lymphatic tissue as a nodule or follicle. Lymphatic nodules can be classified as primary and secondary. If the nodule is dark-stained because it is composed of densely packed lymphocytes, you will refer to it as primary lymphatic nodule. Most likely, the lymphocytes in the primary nodules have not yet encountered their antigens. Once they do, the central portion of that nodule will become light-stained, and it will now become the secondary lymphatic nodule or secondary lymphatic follicles. Take a look at the nodule pointed by the blue arrow. It's dark stained, and this is an example of a primary lymphatic nodule. This is another picture of a primary lymphatic nodule. The nodules shown in this picture are both primary type. But if the nodule has a light staining center surrounded by a dark staining periphery, then you will refer to that nodule as a secondary lymphoid nodule or secondary lymphatic nodule. The light staining area within the secondary nodule is called the germinal center. Majority of the cells that we can find in the germinal center are B cells, and these B cells are activated ones, and they are the ones that will form the memory cells and the plasma cells. And since the plasma cells would use their DNA so that they can produce antibodies, their DNA will be loosely attached to their histones, making their nuclei appear lighter. That's why the central portion of the germinal center is lighter in staining compared to the peripheral portion. Now, the dark staining peripheral portion is referred to as mantle zone or corona. This is a good picture of a secondary lymphatic nodule. Please appreciate the light staining germinal center and the dark staining corona or mantle zone. The lymphatic nodules labeled as number 2, 3, and 4 are all secondary type of nodules. If the diffuse pattern of lymphatic tissues are found in the organs that belong to the gastrointestinal tract, genitourinary tract, and respiratory tract, and they are found below the epithelium, where can we find lymphatic nodules? They can be found in the tonsils, the Peyer's patches of the ileum, and in the appendix. You have learned a while ago that there are 3 types of tonsils. You have palatine, you have lingual, and you have pharyngeal tonsils. This is the cut section of the pharyngeal tonsil. Please appreciate that you have there lymphatic nodules, particularly secondary type. Lymphatic nodules can also be found in the Peyer's patches of the ileum. Let me first help you how to identify ileum under the microscope. Ileum is one of the segments of the small intestine. Small intestine is known to have finger-like projections on its lumen, and these finger-like projections are called villi. So expect to find villi on the lumen of the ileum. And then the villi is always accompanied by a short invagination, and we call that short invagination as the crypt of Lieberkühn. This is a cross-section of the ileum. This is one of its villi. This is one of its crypt of Lieberkühn. This is another villus, and this is another crypt of Lieberkühn. So that will tell you that you're looking at one of the segments of the small intestine. And then take a look at the layer below the villi and crypts of Lieberkühn. Can you appreciate lymphatic nodules? Those are now the Peyer's patches. This is another picture of the ileum. Please appreciate the presence of villi and crypts of Lieberkühn, and take a look at the arrangement of the lymphocytes in the Peyer's patches. They are in nodular pattern. Lymphatic nodules can also be found in the appendix. Appendix is part of the large intestine, and large intestine does not have villi. In contrast to small intestine, the crypts of Lieberkühn of the large intestine are quite longer. This is how the appendix would look like under the microscope. Please appreciate the absence of villi and appreciate the long crypts of Lieberkühn. Can you appreciate that there are lymphatic nodules within the appendix? This is another picture of the cross-section of the appendix. This is one of the long crypts of Lieberkühn. And then take a look at the area below the pointed crypt of Lieberkühn. Can you appreciate a lymphatic nodule? Let us now try to discuss the microscopic features of the different major organs of the lymphatic system. Let's start with the bean-shaped lymph node. You have learned a while ago that it has two surfaces. You have the concave, which contains the hilum, and the convex, which has the afferent lymphatic vessels. Lymph nodes are secondary lymphoid organs, so expect that it will act as a storage of mature T cells and B cells. It will also serve as the site where the T cells and B cells will encounter their specific antigens. And as you have learned from the introduction to this chapter, the lymph nodes can also filter microorganisms and antigens in the lymph introduced via its afferent lymphatic vessel. Lymph nodes are small encapsulated kidney bean-shaped organ, and it has two types of lymphatic vessels. You have the afferent lymphatic vessels present on its convex surface and efferent lymphatic vessels present on its hilum or concave surface. Just like the other organs of the lymphatic system, the lymph nodes have parenchyma and stroma. The parenchyma would always involve the functional cells. We have the T cells and B cells. And for the T cell to be activated, for it to activate B cell, you must also have the presence of antigen-presenting cells in the form of dendritic cells or macrophages. The stroma of the lymph node includes the capsule, trabecula, and reticular fibers. Let's have a review. This is the capsule of the lymph node. The invagination of the capsule will form the trabecula or trabeculae. And then, the cells within the lymph nodes are supported by the reticular fibers. Lymph nodes have two important regions, the cortex and the medulla. The outer dark staining region of the lymph node is called the cortex. I intentionally put the red arrow on the cortical region of the lymph node. Blue arrow is pointing to the capsule, white arrow is pointing to one of the trabeculae, and the red arrow is positioned on the cortex region of the lymph node. The cortex can easily be identified because it is the outer layer and it is dark staining. The cortex of the lymph node is further divided into superficial and deep. Please refer to the blue arrow. The blue arrow is situated on the superficial cortex of the lymph node. That particular area in the lymph node primarily contains B cells, and the B cells are arranged in lymphatic nodular pattern. Take a look at the picture of this lymph node. Immediately below the capsule, you have the superficial cortex. Please appreciate that the cells in there are arranged in nodular pattern, and majority of the cells in the superficial cortex are B cells. Red arrow is showing us the cortex region of the lymph node, while blue arrow is showing us the superficial cortex. The other portion of the cortex of the lymph node is called the deep cortex or paracortex. Please refer to the brown arrow. And in this particular area of the cortex of the lymph node, the cells are arranged in diffuse pattern. Superficial cortex, nodular. Deep cortex, diffuse. Majority of the cells in the superficial cortex are B cells. In the case of deep cortex, majority of the cells are T cells. Red arrow is showing us the whole cortical region of the lymph node. Blue arrow is showing us the superficial cortex. Take note of the nodular patterns of the cells in there. Majority of the cells in the superficial cortex are B cells. Brown arrow is showing us the deep or paracortex, wherein the cells are in diffuse pattern and majority of them are T cells. The inner region of the lymph node is called the medulla. Compared to the cortex, medulla is light staining. And The tissues here are arranged in medullary cords separated by medullary sinuses. I will explain this one later on. Red arrow is showing us the whole cortical region of the lymph node. Green arrow is showing us the light staining medullary region. This is the superficial cortex. This is the deep cortex. The cells in the medulla are arranged in medullary cords. When you say medullary cords, the cells are arranged in a tube-like pattern. These medullary cords are separated by spaces, and these spaces will be referred to as the medullary sinuses. B cells predominate in the superficial cortex. The deep cortex has abundant T cells. The cell that will predominate in the medullary region of the lymph node is the macrophage. What do we know about macrophages? They are phagocytes and they can act as antigen-presenting cells. So if you have them in the medullary cords, then expect that the microorganisms and antigens brought by the lymph into the afferent lymphatic vessel, then into the lymph node, will be captured in the medullary region because you have there the phagocytes, the macrophages. Bacteria will be carried by the lymph and will enter the lymph node via the afferent lymphatic vessels. How does the lymph reach the medulla from the convex surface of the lymph nodes? From the afferent lymphatic vessels, the lymph is drained towards the space below the capsule, and we call that as the subcapsular sinus. From the subcapsular sinus, this lymph will flow into the spaces within the trabeculae, and we call them as the trabecular sinuses. Trabecular sinuses are also known as the cortical sinuses because they are found within the cortex of the lymph node. From the trabecular sinuses, the lymph now will be drained towards the medullary sinuses. The afferent lymphatic vessels will drain on the spaces below the capsule, and we call them as the subcapsular sinuses. Then, lymph will flow through the trabecular sinuses, the sinuses that we can find along the trabeculae of the lymph nodes. But I want you all to think that the cells that we can find on the superficial cortex are B cells, and they are not phagocytes. So therefore, from the subcapsular sinus down to the trabecular sinus, the microorganisms will not yet be attacked by the cells. Along the trabecular sinuses, the microorganism will go down the deep cortex, and you have their T cells. And T cells cannot also recognize these microorganisms and antigens. It is only when the microorganisms and antigens will reach the medulla, that's the time that they will be captured and phagocytosed by the macrophages there. So, understanding the flow of lymph into the lymph node, you will really appreciate that the lymph will go out of the afferent lymphatic vessels microbe-free or antigen-free. Now, what is the reason behind the arrangement of the cells in the lymph nodes? Why do we have B cells on the superficial cortex, T cells on the deep cortex, and macrophages in the medullary cords? Let's have a review of what you have learned in the Introduction to Immunology. Microorganisms will be engulfed by APCs, antigen-presenting cells such as macrophages, and these macrophages will present the antigen to T helper cells. And once activated, the T helper cells will activate B cells to become plasma cells, and the plasma cells will produce antibodies. That is basically the reason behind the arrangements of the cells in the lymph node. The macrophages are found in the medulla because that is where filtration of the lymph happens. So most likely the microorganisms and the antigens will be captured by the macrophages there. Macrophages act as antigen-presenting cells, and they present the antigens to T cells. So might as well place the T cells beside these macrophages, And the best location is the deep cortex or paracortex. And once activated, these T helper cells are supposed to activate B cells. So might as well place B cells beside the T cells. So that's why the cells that will dominate on the superficial cortex is the B cell. You have learned a while ago that lymphocytes can enter the lymph nodes via two means. Number one, via the afferent lymphatic vessels present on the convex side of the lymph nodes. And number two, via the high endothelial venues. And since at this point you can already identify the regions of the lymph nodes, it's now time to identify the location of the high endothelial venues. They are particularly found at the deep cortex of the lymph nodes as shown in the picture. These are two high endothelial venues, so expect that the majority of the cells around them are T cells because they are found in the deep cortex of the lymph node, which is primarily composed of what cell again? T cell. The next organ that we will discuss is the spleen. This is the largest among the lymphatic organs, and it is situated on the upper left quadrant of the abdomen. The spleen functions to, number one, filter the lymph, and this is carried out by its white pulp. And spleen is known to be the graveyard of old red blood cells because one of its function is to destroy old and damaged red blood cells, and this is carried out by its red pulp. This is a fresh cut section of a spleen. The red colored areas are the red pulps, and that is where the old and damaged red blood cells are destroyed. The white circular areas are the white pulps, and that is where the lymph is filtered. This is how the spleen will look like under the microscope. Appreciate that the white pulps appear as lymphatic nodules, and these white pulps are surrounded by the red pulps. For you to easily identify that this is the spleen and not the lymph node, please check the distribution of the lymphatic nodules. In the lymph node, the nodules are found at the superficial cortex, while in the spleen, the lymphatic nodules are scattered all throughout the organ. Again, the lymphatic nodules of the lymph nodes are found on their superficial cortex or below their capsule, while the lymphatic nodules of the spleen, or also known as the white pulp, are distributed all throughout the organ. Just like the lymph nodes, spleen also has parenchyma and stroma. In terms of parenchyma, you have the T cells and the B cells plus the macrophages. And in terms of stroma, the same structures. You have the capsule, trabeculae, and the reticular fibers. This is a cut section of the spleen. Please take note of its protective covering, that's the capsule, and take a look at the extension of the capsule into the organ that is called as the trabecula, singular, trabeculae, plural. Take a look at this nodule that is viewed under high power objective. There's no way for you to identify whether this lymphatic nodule is from a lymph node or a spleen, because as you have learned a while ago, you can simply identify the spleen and the lymph node based on the location of their lymphatic nodules. And that is only possible if you're using the scanner or the low power objective. In this case, how will you know if the lymphatic nodule you're looking at under high power or oil immersion is from a lymph node or a spleen? The answer is the presence of the artery. Can you appreciate the artery in the lymphatic nodule? That is a central artery, and that is only present in the lymphatic nodule or white pulp of the spleen. Take note, even if it is eccentrically located in the white pulps of the spleen, it's still referred to as the central artery. So that's your giveaway clue. The presence of central artery will identify the lymphatic nodule as white pulp, and therefore you're looking at a spleen. Take a look at this lymphatic nodule. Can you appreciate that it has a central artery? So that's a giveaway clue. You're looking at one of the white pulps of the spleen. The lymphocytes encircling the central artery are referred to as the periarterial lymphatic sheath. So the white pulps of the spleen have two parts. You have the central artery and you have the periarterial lymphatic sheath. This yellow arrow is pointing to the red pulp. You have there abundance of red blood cells. This is another picture of the white pulp of the spleen. You have here the presence of the central artery. This is a cut section of the spleen. You can see the capsule on the left side of the photo, and you have nodules distributed all throughout the organ. And the nodules contain central arteries. In the red pulp of the spleen, you have there red arrow, splenic cords, which are basically composed of macrophages. And these macrophages are just waiting for any red blood cell that will be trapped along with the green arrow sinuses. These sinuses are too small that the red blood cells have to deform themselves in order for them to squeeze through the sinuses and go out of the spleen. Old red blood cells are no longer deformable, same as with damaged red blood cells. So what will happen? If these red blood cells will flow through green arrow, splenic sinuses, they will be most likely trapped, and the macrophages in the red arrow, splenic cords, will engulf them and destroy them. By the way, the other name for the splenic cords of the spleen is Billroth cord. This is another picture of the red pulp. The arrows 1, 2, and 3 are all pointing to the splenic sinuses where the red blood cells are supposed to flow through to go out of the spleen. And these splenic sinuses are separated by the Billroth cords or the splenic cords. Sinuses are vascular spaces, and since they are vascular spaces, they are lined by endothelial cells. The blue arrow is pointing to one of the sinuses in the spleen. As you can see, this sinus is lined by these long endothelial cells. These long endothelial cells are also called as stave cells. S-T-A-V-E, stave cells. So I want you to take a look at the photo on the right side of the screen. Take a look at the arrow number 4. The cells beside number 4 are stave cells. They are the ones lining the walls of the splenic sinuses. Let's discuss the circulation of blood in the spleen. You have here the main arterial blood supply of the spleen, that is the splenic artery. The splenic artery will enter through the hilum of the spleen. This is the capsule of the spleen. This is the extension of the capsule into the spleen. We call that as the trabicula. This is one of the small branches of the splenic artery. Do you know that the branches of the splenic artery will send branches that will course through the trabeculae of the spleen? And these branches will be referred to as the trabecular arteries. At some point, these trabecular arteries will produce branches that will go out of the trabecula. And these branches will be covered by or surrounded by lymphocytes. And that is now the central artery that we can find in the white pulps of the spleen. Again, the central artery is that branch of the trabecular artery that goes out of the trabecula and is surrounded by lymphocytes. The lymphocyte surrounding the central artery is referred to as the periarterial lymphatic sheath. After coursing through the white pulps of the spleen, the central arteries will give rise to straight arteries, and these straight arteries are called as the peninsular arteries. Penicillar arteries. These peninsular arteries will eventually form small capillaries. There are two types of circulations in the spleen. If the capillaries from the peninsular arteries will drain directly into the venous drainage of the spleen, you call that as the closed circulation. The other one involves the drainage of the capillaries directly into the red pulp. This is to allow some of the damaged and old red blood cells to be phagocytosed and cleared in the red pulps of the spleen. Then, the blood will eventually drain into the venous drainage of the spleen. I hope at this point you can already differentiate the closed blood circulation and the open blood circulation in the spleen. Let's have a review. This is one of the trabecular arteries. It will send branches that will go out of the trabecula and will be surrounded by the periarterial sheath, and that is now the central artery. The central artery will course through the white pulps and eventually will give rise to the peninsular arteries. Peninsular arteries will form small capillaries. If the small capillaries will drain directly into the venous drainage of the spleen, you call that as the closed circulation. But if the capillaries will drain immediately into the red pulp, allowing some of the damaged and old red blood cells to be cleared from the blood by the splenic macrophages, then you call that as the open circulation. The last but not the least, the thymus. This is a bilobed organ found at the mediastinum of the thoracic cavity, and this organ primarily serves as the site for maturation of T cells. The thoracic cavity is composed of smaller cavities. You have the pericardial cavity, which houses the heart, the pleural cavity, which houses the lungs, and the yellow-colored mediastinum. And that is where we can find our thymus. In terms of the stroma, you have learned a while ago that in the lymph nodes, you have there the protective covering in the form of capsule. Once the capsule will invaginate the lymph node, it will form the trabeculae. In the case of thymus, instead of forming trabeculae, the capsule will give rise to septum. What is now the difference between a trabecula and a septum? Remember, in the lymph node, the capsule will form small invaginations, and these small invaginations will be referred to as trabeculae. In the case of thymus, you have there the capsule, and once the capsule will invaginate the thymus, it will actually divide the organ into lobules. As you can see, you have here one lobule, and this lobule is separated from this lobule, and this lobule is also separated from this lobule. So, the part where the capsule will invaginate the thymus, you will not call it as trabecula. You will call it as a septum, because a septum is supposed to separate things into compartments. This is one thymus, and appreciate the individual lobules that were formed by the septum that came from the capsule. So, the stroma of the thymus should include the capsule, the septum, and the reticular fibers. I want you to focus on one of the lobules. Can you appreciate that each of the lobule has a dark staining outer cortex and a light staining inner medulla? So, the lobules of the thymus would appear like that of the lymph nodes. So, how will you identify which is which? This is a cut section of one of the thymic lobules. Refer to the dark staining cortical region. The cortical region of the lymph nodes also appear to be dark stained. So how will you know whether the organ you're looking at is a lymph node or a thymus? Remember, in the superficial cortex of the lymph nodes, the lymphocytes are arranged in nodular patterns. Take a look at the lymphocytes in the cortical region of the thymus. The lymphocytes are arranged in diffuse pattern. So there is no nodular pattern of arrangement of lymphocytes in the thymus. Then, refer to the light staining medullary region of the thymus. One of the important things that you can find in the medullary region is the presence of the Hassel's corpuscles. Speaking of the active cells of the thymus, we have two. We have the T lymphocytes. You can also refer to them as thymocytes because they are the cells of the thymus. And we have the epithelial reticular cells, and there are 6 types of them. There are no B cells in thymus because the main role of the thymus is to serve as site for maturation of T cells. Aside from the T lymphocytes, there are epithelial reticular cells in the thymic lobules. And there are 6 types of them. So let's start with type 1. Type 1 epithelial reticular cells are found along the capsule and septa of the thymus, and they function to separate the T lymphocytes in the thymus from the capsule and the septa. There are blood vessels flowing through the capsule and the septa, and these blood vessels might carry toxic chemicals that can destroy the lymphocytes. And the function of these type 1 epithelial reticular cells is to protect the T lymphocytes from the harmful chemicals in the blood vessels in the capsule and the septa. These cells are provided with tight junctions, and you know very well that tight junctions do not allow chemicals to pass through between the cells. So therefore, these cells can protect the developing lymphocytes in the thymic lobules from the toxic chemicals present in the blood vessels in the capsule and septa. Therefore, the type 1 epithelial reticular cells are the ones responsible for forming the blood-thymus barrier. Shown here is a picture of a blood vessel. This blood vessel could be present in the capsule or septa of the thymus. What are now the components of the blood-thymus barrier? So let's discuss them one by one. It includes the endothelial cell of the blood vessel. This endothelial cell is resting on its basement membrane. Beneath the basement membrane, you have there a thin perivascular tissue space. Then, I want you to take a look at the basement membrane wherein the type 1 epithelial reticular cell is resting onto. So, these are the 5 components of the blood-thymus barrier. So, let's apply the concept on this particular picture. First is the endothelial cell of the blood vessel, then its basement membrane. Then you have here a thin perivascular tissue space. This is the basement membrane of the type 1 epithelial reticular cells of the thymus. These 5 components are making up the blood-thymus barrier. The type 2 epithelial reticular cells are found within the cortex of the thymic lobules, and they are the ones responsible for facilitating the maturation of T cells. Remember, T cells are produced in the bone marrow, and they will migrate towards the thymus for maturation. And what will help them in the process are the type 2 epithelial reticular cells. Type 3 epithelial reticular cells separate the cortex from medulla, and for them to do that, they are provided with tight junctions. What will separate the medulla from the cortex is the type 4 epithelial reticular cells. Similar to type 3, they are also provided with tight junctions. Type 5 epithelial reticular cells are found within the medulla of the thymic lobule, and they are responsible for organizing the T cells in there, thereby providing support. The last but not the least, the type 6 epithelial reticular cells. Similar to type 5, they are found within the medulla, and they are the ones responsible for the production or synthesis of the Hassel's corpuscles. Remember, these Hassel's corpuscles are characteristic features of the thymic medulla. This is a picture of one of the Hassel's corpuscles in the thymic medulla. It is actually composed of type 6 epithelial reticular cells that are arranged in concentric manner. This is another picture of a Hassel's corpuscle. If this will come out in your exam and you are asked to identify the organ, you should write down on your paper thymus. If you are asked to be specific with your answer, you write down medulla of the thymus or thymic medulla. Please take note that after we reach the age of puberty, our thymus will reduce in size because it will undergo involution. And most scientists are suggesting that what is causing the thymus to atrophy is the reproductive hormones such as estrogen and testosterone. Majority of the areas in the thymus will be replaced by adipose tissue. So let's try to compare the thymus of a child, left photo, and thymus of the adult, right photo. Can you appreciate that on the thymus of adults you have there adipose cells replacing majority of the parenchyma of the thymus? This is Dr. Francis Ian Salaver, and I would like to thank you for taking time to listen to my lecture on histology of the lymphatic system. Good day.